Good morning, everyone. Let's get started. So today I aim to finish lecture 6, and hopefully we can start a little bit of lecture 7 as well. Um, the way things are looking right now, we probably have to postpone the quiz number 3. I will let you know when more people come. Okay. So last class, we started a note on the cell cycle, and we talked about how the cell cycle is separated into two distinct phases. We have the interphase, which is where the cell will spend roughly 90% of its time in. And then we have the mitosis, which is about 10% of the time. And during the interphase, the cell is basically growing, carrying out its normal function, and they are preparing for the actual division, preparing for mitosis. Right? So the interface itself is separated into subsections, if you would. We have the G1, which is primarily growing. Okay, this G1 is the first stage after a cell has finished dividing. Okay, so at that point is quite small. It's not gonna, divide again anytime soon. It's just growing, performing its normal function. And then after it grows for a while, when it gets too big, then it's time to prepare for another round of division. Okay. And as you learned from me last time, before a cell can divide, it must first copy all of its genetic material. It must first undergo DNA replication. And so towards the end of the G1, there is a G1 checkpoint, right? Right here. We took a note on this last time. The G1 checkpoint is to make sure the cell is ready to copy its genetic material. And so several things are being checked at this checkpoint, right? Do you guys remember any of it? Maybe from the notes, you can take a look. What are we checking for to make sure the cell is ready to copy its genetic material? I'm sorry, what's that? I just didn't hear you. No, okay, that's okay. Anyone else? What do we need to make sure? What's that? Yeah, the quality of DNA, right? We want to make sure the quality of DNA is good, that there are no damage, right? And if there are damages, you need to try to repair it, you try to fix it, right? You need to make sure the telomeres are long enough, right? It's not critically short. You need to make sure you have the resources to actually go on and do the DNA replication, right? Do you have enough nucleotides, right? So there are all these things that you need to check. And if the cell passed the checkpoint, then it's allowed to move on to the S phase, where it will grow some more, and at the same time, it will copy its DNA. Right? And the way it copies the DNA is the long note that we took, right? With the gyrase and the helicase and all those things. If it does not pass the G1 checkpoint, then it's going to be triggering apoptosis. Right? If there are irreparable damages, then you're going to cause the cell to die. It might remain specialized and not divide anymore. It's like, you know what, the telomeres are too short. Let's not go through another round of division. But you can keep on doing whatever cellular function you were doing, right, before. You can continue to work, just that you're not allowed to divide, right? And then, if everything is okay, though, it will go through the production, DNA replication. After DNA replication, there's going to be another checkpoint here, right? This is G2 checkpoint. And you are basically checking to see if the DNA you've copied are of good quality, right? Did you make any mistakes while you're copying it? Did you damage DNA? Right? And again, if there are damages and mistakes, you try to fix them. And if you cannot fix them, you're gonna stop the cell from proceeding further. But if all is good, then it's going to grow some more, and then it's going to prepare for the actual division. So the actual cell division is called mitosis. Right? We learned about the purpose of mitosis for growth and development, for replacing lost or damaged cells, and for some organism, mitosis is a way for reproduction. Right? Of course, that's asexual reproduction. Right? A bacteria can split and create more copies of itself. Right? That's an example of asexual reproduction, although it's not quite mitosis that they do, but it's similar concept. And as you can see, in mitosis, there are different stages, which we're going to look at in more details in the upcoming slides. So that's where we left off last time. Here comes the new stuff. Oh, no, not yet. Sorry, another slide, right? The cell cycle checkpoint, right? This is just basically what I told you. It's the cell's attempt to identify any issues before allowing the cell to divide. And this is an important thing to keep in mind. We're actually going to come back to this towards the end of the lesson. If these checkpoints fail and you allow the cell to divide despite having errors and mistakes, then those mistakes and errors are going to persist in all the new cells that you get from that point forward. Okay, let me show you this in a picture. You don't have to copy this down. I'll give you like formal notes later, but basically it's like this. Okay, let's say this is the original cell, all right, and then it's gonna divide. Right, so you're gonna get like two cells here, and then let's say it's gonna divide again. Okay, but this time when it divides, it actually made a mistake during DNA replication, and let's say those mistakes were not corrected, then what happens is you're gonna get these different cells, okay? They might still be functional, but now it's different. It contains the mistake. And every single cell after that is gonna carry the same mistake. Do you understand? Okay. So what we will learn later is when this process starts, sometimes it's a snowball effect, meaning it gets bigger and bigger as it rolls down the hill, right? In the context of this, it will accumulate more and more mutation, right? You make one mistake, and then now you continue to make more mistakes. And then after several rounds of division, you might end up with something that's very different genetically compared to the origin. It might not happen, but it could happen. You understand? You guys might have like a mole somewhere in the body. Like I got a mole like here and there. A mole is basically this situation. Okay, like your skin cell divide. And then somewhere along the way, it made a mistake and it changes the color. Okay, so for like a mole is usually darker in color, right? Okay, and so that cell with a mistake continues to grow and grow and grow. And now you have a group of cells that's basically making up the mole, right? It's different from the surrounding cells because this cluster of cells all have the same mistake. Does that make sense? Okay, and as time goes on, that's why people say you gotta monitor your moles, right? Like if it changes color, if it's irregular in size, right? If the edge is kind of not well defined, then it might be like skin cancer, right? And that's what I was trying to tell you. Once you start this process of mutation, there's a chance that it will accumulate even more mutations, and eventually it changes the cell and makes it cancerous. Okay. All right. Stay with me, guys. Exactly a quarter of you are paying attention there. That's after 75 is just like staring into emptiness. Like, what's going on? I can't hear a word he's saying. Okay. But please pay attention because this is difficult stuff to learn on your own. Okay. All right. Let's go back to here. So I am going to go through the mitosis process with you. There are some steps involved. I will not ask you to describe mitosis on a test or quiz, but you have to know what happened in each step to answer some of the questions, okay? So it would be like a multiple choice question. I might give you pictures to organize or identify, but I will not ask you to describe it, okay? So that should give you some relief, perhaps. Okay, I gotta turn off this lights a little bit for this part here because it's hard to see the screen, but don't fall asleep. Yeah, I gotta turn those off too, I guess. All right, so this is not mitosis, huh? This is still interphase. All right, this is an actual picture of a cell that is an interphase, and then later on you will see pictures of cells that are undergoing mitosis. And there are three colors here, right? There are different techniques you can use to color different parts of the cell. In this case, the blue is going to highlight the DNA as well as the nucleus, basically, okay? The greenish yellow thing here, those are the cytoskeleton. Now, specifically, what I need you to recall from earlier lesson is that during cell division, the cytoskeleton that's responsible for moving the chromosomes around, they are called the spindle fibers. Okay, so let me write down for you. I'll try to use the same color here, I guess. So the blue that you see, that's where the DNA are. Okay, DNA in the nucleus, of course. And then we have the greenish thing, yellowish things. I'm just gonna say green. Those are the spindle fibers. Spindle fibers. And the red is just basically the cytoplasm. Okay, so that's what you're looking at. Okay. If you forgot what spindle fibers are, they are produced by these centrioles, and the job is to guide the chromosome movements. Okay. So I want to ask you, do you see the DNA in this picture? It's not a trick question, okay? Just answer, like, instinctively. Yes, you see it, or no, you don't see it? Yeah. Okay, I don't see anything. So very good if you see something. The correct answer is no. I don't see anything. What are you guys talking about? It's just like a blue circle, right? Okay. It's like those psychology tests, right? They show you a picture with nothing in it. They say there's like a zebra in it, and you're like, oh yeah, I see a zebra. This is one of those things. Okay. So look, now let me just move on. Now, do you see the DNA in this picture? And now you can see the individual, like the individual wormy looking things, right? Do you understand what I'm saying? Okay. You can see like almost the little noodles, right? Okay. But here you don't see anything, right? Okay. Now, this is an important point to remember, because when a cell is in interphase, the DNA exists in the form of chromatin. Okay. Do you remember the difference between chromatin and chromosome? No? Chromatin is DNA wrapped around proteins, and that's the normal state in the nucleus. And then when you need to move them around, they condense more to become chromosome. Okay? So when it's not very condensed, it's hard to see it under the microscope. But when they condense, when they pack up, then you can see them individually. Okay, so back to this picture here. When it's interphase, we can't really see the DNA. Okay, they still soak up the blue dye. Okay, we know they're in the nucleus, but we cannot make out the individual DNA. But when we start the mitosis, when we start the actual division, you can see that they become more well-defined. They are more condensed now because they convert into chromosome. Okay, so that's what I wanted to show you with the lights off. Let's bring them back on. Okay, so let's go back to here. Interphase. As I told you, the cell spends most of its life in this stage. Okay, and then it performs regular function. But near the end of this phase, the cell prepares to undergo mitosis. That's the G1S and the G2. And then the DNA duplicates itself. Okay. In this cartoon picture, you can see the chromatin. Chromatin is basically DNA wrapped around proteins. Okay. Not visible. Not clearly visible, I guess. Clearly visible under microscope. Okay. It's just like a hazy fuzz. Right? You can't make out the individual chromosomes. We have 46, right? Okay, but now it's just like a tangled mess. Can't really see anything. And of course, they are in the nucleus. Right, so if you cannot read the writing, it says DNA wrapped around proteins. Sorry, not clearly visible under the microscope. Okay, not clearly visible under the microscope. You know it's there because it soaked up the blue, but you can't see the individual. Think of it like this, like a large crowd at a concert, and then you like a bird's eye view from the sky. Like those are people, but you can't see the individual person. Okay, it's just all mingled together. That's what these chromatin is like. All right. So assuming everything is good, you copy the DNA, no mistakes, no errors, no damage, then we are going to move on to the actual mitosis. The mitosis itself is separated into 4 stages listed here. You can remember PMET. Okay, PMET. That's the order it happens in. Prophase, metaphase, anaphase, and telophase. Okay? And again, I'm not going to ask you to describe them on the test, but you need to know what's happening in each stage. Let's start with prophase. Okay? So we have early prophase and later on prophase. But P, you can remember, P is for prepare. Prepare. You've already prepped some during interface, but let's prepare more. Okay? So in this case, the chromatin, which again is less condensed version of the DNA, they are going to coil into visible chromosomes. You're going to pack it up even more. So that's why now you can start to see the individual outline of the chromosomes. Right? By this point, DNA replication is already completed. Okay, so you've copied all 46 chromosomes. Give me a second. Let me just jump ahead and see. Okay, so I forgot to include a slide here, but I need to explain this to you because it's a little bit confusing. So class notes 203, November 1st. All right. So there are some terms I need to go through with you. All right. If you have colors, please use them. Now, at this point, you should understand we have 2 sets of chromosome, right? 1 set from each parent. And this is chromosome 1 from mom and chromosome 1 from dad. Right? They are the same size, same length, but the genetic information on them might be a little bit different. Okay, I'll explain more about this later. But for example, over here, you might have a gene for brown eyes because your father has brown eyes. And then you might have a gene here for blue eyes. Okay, they are both eye color, but then the version might be a little bit different. Okay, so these are not identical chromosomes, yet they contain the same kind of information. So if you find eye color here, you will find eye color here. If you find a blood type here, you will find blood type here. Okay, but then there are different versions of each information, right? Okay, hair type, like curly hair versus straight hair. Okay, so this is chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, 3, 3, 4, 4, 5, 5, all the way to what number? 23, 22, right? Okay, the 23rd pair is the sex chromosome XX or XY. Okay, XX are same length, obviously, but Y is much shorter than the X. Yeah, we're gonna get more into that later. Right? So for simplicity, I'm not gonna draw all of them. Okay. So here's a question. It's not a trick question, just answer directly. And it's an important question. That's why I asked it out loud. Okay. How many chromosomes do you see on the screen? 6, right? Okay, that's good. So of course there are like 46 in total, but I'm only gonna draw 6 here. So after DNA replication, right, this is before, and then we're gonna go DNA replication. After DNA replication, this is what's gonna happen. Okay, you will have chromosome 1, but then now you will have a copy of it. Right, chromosome 2, you will have a copy. Chromosome 3, you will have a copy. Same thing over here. So these two things are identical copies. Right? These two are identical. Right, so on and so forth, right? So now you copy everything. I want to ask you again. Take a look here, please. How many chromosomes do you have now after DNA replication? Yeah, so like instinctively, you would think it's 12, right? Because now you copied it, right? Okay. But here's the thing that confuses people. If they are stuck together, we still count them as one. Okay. That's just like whoever came up with this concept decided that's the way to do it. So even though there are like two pieces here, but they're stuck together, so we still count that as one chromosome. Okay. So you started with 6 in the picture. After DNA replication, you still have 6. 1, 2, 3, 4, 5, 6. Do you understand? Okay. So when do you count them as 2? When you separate them. Okay. You guys know Kit Kat, like the chocolate? Yeah. So like my kids, they got a lot of Kit Kat from Halloween, right? And so, I don't want them to eat too much. So I separate the bar and I give one to each of the daughter. And then the younger was like, I want 2. And then so I took her bar and then I snap it in half. And it's like, now you have 2. Okay. So if it's together, it's 1. But if you break it, you can count them separately, right? Okay. Now, over here, where they are stuck together, that is called the centromere. Now, there are a lot of terms, okay? Don't confuse centromere with telomeres. Okay, telomeres is what we learned last class, and they're at the ends, right? Okay, and then this centromere is where the two things are stuck together. Okay. So this is 6 chromosome. This is also 6 chromosome. Chromosomes, I guess. Now, just one more term here. These things that are identical copies of each other, we call them sister chromatid. Okay, so that's sister chromatid. they stick together. Sister chromatids. They are identical copies. Sister chromatids, these are not sister chromatids. Okay? Only if they're stuck together and they're copies of each other. Sister chromatids, sister chromatids. Okay? Non-sister chromatids. These are also not. Understand? So, right? All the confusing words. Chromatin, that's just uncondensed DNA, right? When it's not wrapped up. Chromatid, that's the sister chromatids when they're stuck together. Centromere is where they are stuck together. Centromere, 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 centromere. Chromosome is just same as chromatin, but more compact. Any questions about these terms? Want to chromatin, it's like uncondensed. It's like the yo-yo, remember? Okay, you wrap the string around the yo-yo. Chromatid is identical copy. After DNA replication. And they are stuck together. Chromatid, chromatid, sister chromatids. Sisters stick together. Centromere is location where are attached. And chromosomes are just condensed version of chromosomes. Okay, I'll leave that there for the whole day. You can copy it later. I want to go back to this for the moment, please. Okay, so at early prophase, chromatin coils into visible chromosomes. And you pack it up even more because you're about to move them around in the cell. And think about moving. You got to pack your stuff in a suitcase so it's easier to carry. So you're going to pack them up into condensed version so that you can move them around. DNA replication is completed by this point, right? Of course, it's completed because it happened during interphase, right? Sister chromatids, TID, are held together at centromere. The nuclear envelope, which is the membrane that surrounds the nucleus, starts to disappear so that the chromosome can come out and you can move them around. And the centrioles, which are these organelles, they start to move to the opposite side of the cell and at the same time produce the spindle fibers. Okay, these lines that you see over here, these are spindle fibers. They are going to guide the chromosome movement. As time continues, this is late prophase. Now you can clearly see the individual chromosomes, right? Like these little noodles, gummy worm looking things, that's chromosome. Right? And it's much more clear now because the nuclear envelope is completely dissolved. Right? They're able to spread out. So it's no longer this fuzzy sea of DNA genetic material. They start to spread out a little bit. So they are clearly visible. And then the spindle fibers are going to attach to the centromere of each chromosome. Remember, centromere is basically where they're attached to it. And you can't really see it in the picture, but in this cartoon, you can see it. Right, so that's prophase. Right, you're just getting everything ready so that the cell can split. All right. Next is metaphase. M is for middle. That's how you can remember it, middle. Each chromosome is going to experience a tug of war, right? The spindle fiber is going to pull it back and forth until all of them lines up in the middle of the cell. Right? So we have the chromosomes all lined up. Of course, in an actual cell, you will have 46 of these things, right? But for simplicity, we're showing 4. And so they all line up in the middle. That's it for metaphase. M for middle. Okay. What comes after metaphase? PMAT, right? PMAT. So after metaphase is anaphase. A for apart. This is where the sister chromatids get pulled apart. Right? Pull them apart so it's separated. Remember, this copy is exactly the same as that copy, right? They were stuck together before, but now you separate it. So how many chromosomes in this picture now? 8, right? Okay, so in metaphase, it was still 4, even though we already copied it. But now, you break the Kit Kat, you separate the sister chromatids, all of a sudden you have 8. Okay? With real numbers, 46 now is 92. Okay? Remember, you only count them as individual things if they're not stuck together. Finally, telophase and cytokinesis. So basically, it's like this, right? The next phase is to divide the cell. So each half of the cell, each new cell will get exactly one set of the of like the separated stuff, right? So here, telophase, T for tearing, herring, right? You literally will tear the cell into 2, and everything is reverse of prophase. Right? I'm just going to read it to you. Chromosome uncoil back into chromatin. You finish moving them. You can unpack now. You're going to form the nuclear envelope again. We don't need the spindle fibers anymore because we're done moving chromosomes. And then the cell will split in the center by a process called cytokinesis. Cytokinesis is basically the division of the cytoplasm. You have to make sure each daughter cell gets the same amount of cytoplasm. Okay. Otherwise, if one gets more, the other gets less, the smaller cell might not survive, right? So that's what cytokinesis is going to happen at the end of telophase, and it separates the cell. So that's the division. Okay. All right, let's look at some of these pictures. Is it this button? Yeah. All right. Do you guys know what these what cell these are? I mean, it's in plants, but do you know what plants this is? I think I might have told you before, right? Onion, that's right. Excellent. So let's do an easy one. What phase do you think this is at? Is it interphase, prophase, metaphase, anaphase, telophase? Which one is it? You know, why don't you guys talk about it? Okay, I'll give you, I don't know, 1 minute, okay? 2 maybe. I talk with people around you, and then I'm gonna take it up with you, okay? All right. What about A? What do you think it is? Just yell them out. Yeah, metaphase, that's good. And what's the telltale sign that it is metaphase? It's right in the middle, right? Okay, all the chromosomes is in the middle of the cell. Okay, plant cells are more rectangular, right? Ours are like circular. Okay, so that's metaphase. How about this one? That is absolutely anaphase. Can we find more anaphase, guys? Yeah? Yeah, like there, right? I can totally see what you guys are pointing at. Um, that's an anaphase, right? Okay. That's another anaphase, right? Okay. I'm just gonna put A here, A here. How about this one? That's telophase, right? So how do you tell the difference between anaphase and telophase? Well, if you look closely, you can see that a new wall is forming, right? Like a new divider is forming here, right? And also for telophase, it is more concentrated, like all more packed together, the chromosomes, than anaphase. Anaphase, you can still see the individual chromosomes, but here they are kind of going back into their individual nucleus, right? Okay, so this one here is definitely telophase. Okay. Now this one is tricky. Do you think this is I mean, what do you think this is? Yeah, I mean there's only one answer left, right? So this is prophase. Okay. But how about this? What do you think that one? About that one? Is that prophase? Yeah, that is interphase. Very good. Okay. How do you tell the difference between interphase and prophase? Well, remember, in interphase, you cannot see the individual DNA, right? They are still in the form of chromatin. If it's chromatin, it's just like a hue, right? Like a sea of DNA. But in prophase, you can start to make out the individual shape. Do you understand? So you might see some of these questions on the quiz, where I give you pictures and ask you to identify. It's gonna be obvious ones. Okay, I'm not gonna try to trick you or anything like that. Okay, that's it. All right, let's do some poll questions. Okay. We do have a lot to cover, so let's go through this thing quickly. What do you think? Good. Anyone else? Yes. Mm-hmm. Mm-hmm. Good. Good. Yeah. Prophase, right? You need the nuclear envelope to be gone before the chromosomes can come out. Okay? So the answer is A. Okay? Circle it. How about this? Anyone else? What is it? Okay, all of except a few people, most of you said 16. In metaphase, have they separated yet? Not yet. If they are not separated, we count them as one, right? But the answer is still 8. Okay. Oh, sorry. I read the question wrong. Okay, my bad. Okay, sorry. The question says the cell has chromosome 8 chromosome and metaphase. How many will it have in anaphase? 16. Sorry. I read the question as it has 8 and how many in the metaphysics. Sorry. You guys can think about this one. I don't have time to go through it. But a lot of chemotherapy. Chemotherapy is therapy where they use drugs to treat cancer, as opposed to like radiation therapy, for example, right? A lot of these chemotherapy use compound that targets rapid cell division. Okay. Because that's what cancer is. I told you it's uncontrolled cell division. And so you want to slow down the division, which is why side effects of a lot of these chemotherapies, like you heard of like people losing hair, right? You heard about people not able to produce new red cells, for example. That's because your healthy cells are often targeted by these compounds as well. Okay. All right. The next thing is going to be quite complicated. All right. So I need you to be very alert for this new thing. But first, I have a few things to tell you. Almost all of you are here today, which is great. So this is the schedule, and we are here right now. Today is the 4th. I feel like we might be able to start 7. I don't know. Okay. But we definitely will on a Wednesday. We were supposed to have a quiz next Monday, but that's not going to happen. Okay. Monday, instead, we will do the lab. Okay. So I have the microscope lab here. Right. I'll give it to you during the break, or you can pick it up yourself, actually. So you do that. You will hand in the lab on Wednesday. Okay. We'll have another lesson on Wednesday. And then I think we will move the quiz to the 18th. Okay, we'll move the quiz to the 18th. All right, so write that down. I will confirm with you after Wednesday or next week. We'll see where we're at, but tentatively 18th. Okay? Huh? We'll worry about that later. But don't miss the lab. Okay, for next Monday, and microscopy lab, which I'll give it to you later. All right, back to this stuff. Now, the cell cycle. Cell cycle, as you know now, involves cell division, right? So if a cell goes through the cell cycle once, it would split once, right? There are checkpoints along the way. We know that already, G1 and G2 primarily, but there are other smaller checkpoints. And these checkpoints are usually regulated by proteins. Okay, like what's actually doing the checking, right? When I say you have to check for the quality of the DNA, you have to check for the length of the telomeres, what exactly is doing the checking? Well, they are actually proteins. Okay, so these proteins help regulate the cell cycle. And there are 2 kinds of regulators. Okay? Like I said, this is very complicated. Please stop me if you have any questions. One type of regulator, they're called growth factors. These growth factors promote mitosis. Right? They encourage the cell to divide. So we can think of them as green lights. On the other hand, we have growth inhibitors. Growth inhibitors, as the name implies, will block the cell cycle. It will inhibit or slow down mitosis. So we can think of them as red lights. So it's an important balancing act for you to have green lights and red lights that lights up at the right time. Okay? If you need more cells, then you turn on the green lights and let them divide. But you don't want it to be green all the time, right? And you need to slow it down in some cases. Otherwise, you have uncontrolled cell division. Do we understand the difference between growth factor and growth inhibitor. Just give me a tiny nod if you're with me. Great. Now, these growth factors and growth inhibitors, I told you, are proteins. So to make them, you need to have a gene, right? Now, in case you don't know what a gene is basically a segment of DNA with instruction to make a protein. So every protein that you make in the body needs instruction on how to make it. That instruction is in the DNA. One gene allows you to make one protein, generally speaking. And as we know, if the instruction is in the DNA, it is possible to make a mistake in the DNA, right? Particularly when you copy the DNA, right? That's not the only time you can make mistakes, right? There are other situations that could cause your DNA to change, right? For example, UV light, right? That's why people suggest you put on sunscreen, because the UV light can directly damage your DNA and cause them to change. Okay, so if we have a change in the gene, that could cause the protein that it codes for to change as well. Do you guys understand that? Okay. The DNA is the instruction to make the protein. So if you change the DNA, you're going to change the protein. Okay? If you change the DNA, you're going to change the protein. So here, proto-oncogene. A proto-oncogene is a gene that normally helps cell grow. In other words, the proto-oncogenes code for a growth factor. Okay. It is the instruction to make the green light, if you would. Okay. When a proto-oncogene mutates, when it changes, or there are too many copies of it, which could also happen, then it becomes bad. Right? And this bad gene is now called an oncogene. Oncogene. Right? People with cancer often see an oncologist. Oncologist and oncogene, same root word, right?
Is it kind of the same as the onset when you have cancer?
Yeah, sort of, yeah.
I got mine, but
To see if, there are certain activators, right?
yeah, if I'm high.
So, right, exactly. So proto-oncogene is the normal version. We all have it, okay? That's just the instruction to make the green lights, the growth factor. When that instruction got mutated, right, then it becomes an oncogene. And we do not want too many oncogenes. Okay, we don't want any, in fact. Okay. So, how is that going to be a problem? Well, if it gets mutated into oncogene, then the gene can become permanently turned on or permanently activated. Okay, what that means is instead of having green lights that turns on and off, under the right circumstances, now the green light is permanently on. So the cell cycle just keeps on going and going and going. Does that make sense? On the other hand, we have the tumor suppressor gene. Tumor suppressor genes are normal genes that slow down cell division. They are the instruction to make the red light. Okay? When a tumor suppressor gene don't work properly, then the cell can also be out of control. The cell cycle can be out of control in one of two ways. Either the green lights are always on, or the red lights are not turning on. Do you understand that? If the green light is always on, then it's just go, 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 right? Divide, 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 divide. If the red lights are not on, then nothing is stopping the cell from dividing. And they will also go divide, divide, divide. Okay. So a mutation in proto-oncogene is a green light that does not turn off. A mutation in the tumor suppressor gene is a red light that does not turn on. Okay? So we call that a gain of function mutation in the proto-oncogene. A gain of function means it is always on. Okay? Here, I'll write down gain of function. Should probably use this bright color as well. Green light is always on. Now we have 2 copies of each gene, right? Because we have 2 copies of each chromosome. Generally speaking, a mutation in one of the proto-oncogene is enough to cause the cancer. Okay. So here is like another example. You have the normal genes, and then the growth is being regulated. But over here, a mutation caused the accelerator to be stuck. So your car is just going to go really quickly with no way of stopping. Okay. In reality, though, that doesn't happen too often. More realistically, you need multiple mutations to get cancer. Okay. But, we're going to keep it simple. So it's like this. Right? You have 2 copies of the proto-oncogene, and when it's functioning properly, like both of them is going to turn on, and then when they are both on, then the cell divides, and when they're both off, then the cell don't divide, right? Okay, both on, divide, both off, don't divide. Get it? Now you have a mutation. One of them is mutated, and it stays on. So if you look at this one is the normal one. Right? It's able to turn off, turn on, turn off, turn on. But your cells cannot pay attention to this one because the other one is always on. So even though your cell sees that, oh, this says stop, but that says go. So let's divide. Okay? That's why if even just one copy of it is mutated, that's enough to cause the cell to have uncontrolled cell division. Okay. On the other hand, for the tumor suppressor gene, right, you have 2 copies of the red light. Okay? And let's say one of them does not work. Right? So like, you try to turn this one off, right? And the cell can still see this one. Right? There's a red light here. Even though I don't see this one, there's a red light here. So what are you going to do? You're going to stop, right? Okay. It's only when both copies are busted, then the cell will not stop. Okay. So there's a difference here. For oncogene, 1 copy is enough to cause it to divide uncontrollably. For tumor suppressor gene, both copies has to be not working for it to cause cancer. And so this is called loss of function in tumor suppressor gene. Both copies of the gene need to be mutated to cause cancer. And so people born with only 1 functional copy are more likely to get cancer. Okay, here is why. This part is a little bit difficult to explain. I'll try to explain it on the board. Okay, so let's say, there are a bunch of cells in the body, of course, trillions of cells, okay? Each of them would have 2 copies of the tumor suppressor gene. Right? So let's say there is a mutation and one of the tumor suppressor gene is busted here. Okay? So how does the person get cancer? They would need to have a second mutation in the same cell, right? Okay. So let's say they have another mutation here. Is that going to cause cancer? No, because they are separate cells, right? Okay. So you need to have the same mutation twice in the same cell in order for it to be a problem. You have to make the red light not work in the same cell twice before it becomes a problem, right? And that's very unlikely. Okay. That's like, me blindfolding myself and then I throw like a softball into the classroom and it hits the same person twice in a row. Okay. Like that's just unlikely, right? However, if a person inherits 1 mutated copies from the parents, then all their cells would have one non-functioning red light already. Okay? And now, if the person has just one mutation in any of the cells, that's enough to push them over the edge and cause them to have cancer.
What if your genes are, there is no mutated gene in your genes?
Well, then it takes two hits, right? And it takes two hits, right? So it's like more unlikely. So that's why, right? Cancer can be inherited in some forms or acquired, means you get it yourself, right? And based on what we discussed, this is the important point. When someone inherited an abnormal copy of the gene, their cell already start out with one mutation. So it's easier to get the next mutation. Right? So cancers that are inherited tend to occur earlier in life than cancer of the same type that are not inherited. Okay? So take breast cancer, for example. If we're talking about the inherited form of it, it's going to likely occur earlier in life because it only needs one more mutation for it to happen, right? Compared to breast cancer in general. Make sense? There is also the idea of penetrance, okay? Penetrance is the concept that refers to what percent of the people with a particular mutation will actually get the cancer, okay? A lot of things is not like for certain. Right? It's not like if you get this mutation, then you're guaranteed going to have a particular form of cancer, okay? So let's say 100 people have this mutation, but only 60%, 60 of them actually gets the cancer. Then we say there's a penetrance of 60%. Okay. Some things have 100% penetrance. It means you're guaranteed to have it if you have this mutation. Some things have low penetrance. Okay. It just increases your risk of getting the cancer, but not necessarily definitively. Okay, so a very common thing for people to do nowadays is to do genetic testing on themselves, right? Like you can actually buy a kit online, and then you swap yourself, and then you send the DNA sample, and then they will analyze your DNA, right, and tell you, what your risks are, what's your expected life expectancy and all those stuff, right? Those things are it's very broad, right? Because in part of the concept of penetrance. Just because you have certain genes doesn't automatically mean you're going to get certain diseases. Okay, so I'm going to go through one more thing and I'm going to give you a break, okay? An example of a tumor suppressor gene is the BRCA1 gene. Right? BRCA1 stands for breast cancer 1 gene. And this gene is responsible for repairing damaged DNA or destroying cells when the DNA cannot be repaired. So this is actually a tumor suppressor gene. Tumor suppressor gene. All right. Because it slows down cell division as opposed to speed it up. Oh, it actually says right here. So it prevents cell division in the event of irreparable errors, and it's considered as a tumor suppressor gene. So how many mutations in the BRCA1 gene would a person need for them to develop the cancer? 1 or 2? 1 or 2. This is the red light, right? Do you need both of them to be not working to have cancer or just one of them? 2 of them, right? Okay, 2 of them. Okay. About 12% of women in the general public will develop breast cancer sometime during their lives. Okay, 12%. Right? And it tends to happen, later on in life, generally speaking. However, 55 to 65% of women who inherit a harmful BRCA1 mutation will develop breast cancer by age 70. Okay? So if they inherit a mutated version of the BRCA1 gene, if the DNA repairing mechanism is not there, or you cannot stop the cell when it has error, right, in all the cells, then the risk of developing the cancer is much higher compared to the general public who does not have a faulty copy. Okay. Some of you might know the actress Angelina Jolie. Her mother died of breast cancer, I think. And so she did genetic testing and found out she inherited the mutated copy of BRCA1 from her mother. Okay. Which means now her risk of getting breast cancer is much higher than otherwise, right? Okay. So for her, as a preventive measure, she underwent double mastectomy surgery. Okay, so she removed her breast just so that she won't get breast cancer. Okay. And she did an interview about that. Okay. So, it actually caused quite a bit of controversies at the time when that happened, because some people are saying, it actually put extra stress on the healthcare system if a lot of people start screening for these things and then taking preemptive surgery, right, to prevent something that might or might not happen. Others are saying, it's an empowering choice, and if people are able to make an informed decision about that, they should be able to do it. Okay, so if you're interested, you can read more about that. Okay, that's 9:30. Let's take a break, and then we'll continue afterwards. Please help yourself with So once the cell has accumulated enough mutations, and like I said, often it's not as simple as I've explained it to be. It's not like, you get a single mutation and then you're gonna get cancer. It's often multiple mutations, but the underlying principle is the same. Okay, it has to do with either overly active oncogenes or not working tumor suppressor gene. Right? It's either the green lights that are always on or the red lights that are not turning on at all. Okay, so if enough red lights and enough green lights malfunction, then the cell is gonna divide in an uncontrolled manner. Right? That's the bottom line thing to remember. And so when a cell keeps on dividing and dividing and dividing, eventually it forms a tumor or neoplasm. Okay, neo means new. It's neoplasm means like a new growth, basically. Okay, that wasn't there before. Now, a tumor is not necessarily cancerous. A lot of time, if a person has a tumor, the doctor will do a biopsy, like a sample of the tumor. Biopsy, biopsy, which means they take a sample of the tumor and then they look at it under the microscope. Okay, a lot of times they stain it and look for features. And if it's benign, then it means it's not dangerous. Sometimes you can remove it with surgeries if it's not in a tricky position, and there will be no further complications. However, if the tumor is malignant, right, then it means it is cancerous and it could potentially get progressively worse, and it's dangerous to health. So you will try to intervene as early as possible with surgery, with chemotherapy, radiation therapy, and more recently, there is something called immunotherapy as well, okay, where you train your cells, your white cells, to look for these tumor cells in the body and destroy them. That's something we'll look at more closely next semester. So what are they looking for in the biopsy to determine whether it is benign or malignant? Well, it turns out there are 6 hallmarks of cancer. Right? These are characteristics that are found in all cancer cells. So let's take a closer look at some of these things. Number 1, self-sufficiency in growth signal. That means because of the mutation in the oncogene, they have their own green lights that are always turned on. They don't need external factors to tell them to divide. They will tell themselves to divide. And this self-sufficiency in growth signal allows them to keep on progressing through the cell cycle again and again and again. To make things worse, they are also insensitive to anti-growth signal. All right. They no longer respond to red lights. Okay? Even though you turn on the red lights and you tell them to stop dividing, they don't listen to it. Okay? They have their own signal to tell them to keep going, and they ignore the signal that tells them to stop. Understand? All right. Next, they are able to evade apoptosis, evade death. Right? Remember, when a cell fails G1 or G2 checkpoint, it's supposed to go and get triggered with apoptosis, right? It's supposed to kill off the cell and not let it go forward. But cancer cells ignore that. They don't die. They essentially become immortal. Okay? Remember I told you about the HeLa cells from way back, lecture 3. They took the ovarian cancer cells from the patient and they're able to grow it continuously in a dish. They become immortalized. Okay, you can't do that with regular cells. Even if you give ideal conditions, best conditions for growth, if you take a regular cells and try to grow it in those ideal conditions, it will stop growing eventually. Okay, but not cancer cells. Next, sustained angiogenesis. Genesis is to create, angio is blood vessels. So basically, they create new blood vessels. Right. And that's an important thing for a rapidly growing tumor. It requires a lot of energy and resources. And so it redirects the resources by growing new blood vessels to supply the tumor. Right? And it essentially leaches the nutrients and the resources from the rest of the body, okay, to sustain the growth. So insensitivity to stop anti-growth signal, self-sufficiency in growth signal, evade apoptosis, create new blood vessels. They also have limitless replicative potential. Okay, they do not become senescent, right, because of telomerase activation. Okay. Normally, when a cell divides so rapidly, its telomere is going to run out, and then the cell becomes senescent. It's not allowed to grow anymore. But they turn on the telomerase in these cancer cells, and so they reset the telomere length regularly. They could keep on going. And finally, the last hallmark of cancer is its ability to invade surrounding tissue and spread to other body sites. Okay, metastasis is spreading. All right, so this doesn't often happen early on. It's only at the later advanced stage that it starts to spread to other places. So there you have it, 6 hallmarks of cancer. Okay, remember a few of them, please, for the upcoming quiz, okay? I don't have time to go through these things, but you can try them later on, okay? I'll probably post the answer for you later on as well. So that's it for lecture 6. All of lecture 6 will be on quiz number 3. Okay, all of lecture 6 will be on quiz number 3. And unfortunately, some of lecture 7 will also be on, okay? Not the whole thing though, right? Not the whole thing. I'll tell you where the cutoff is. But we will start number 7 right now. Seven, 8, and 9 is less about memorization and more about problem solving and applications, okay? So that might be a little bit better for you if you're good with, problem solving, like, math and chemistry, that kind of stuff, right? We'll be doing some problems. Yeah, so it's a nice switch from the stuff that we've been doing. Lecture 7, protein synthesis. Okay? So we now understand DNA codes for the instruction to make the protein, but how exactly do we take the ACTGs and change it into proteins? Okay, so that's what I will teach you. I don't know if we can get to the protein part today. We'll see. Remember the central dogma, central theme, right, of biology, which I've shown you before, starts at DNA, and then you're going to make a copy of DNA in the form of what? Yep, RNA, and then it's going to become protein, right? This part is called transcription. This part is called, anybody? Very good, translation. So we will talk about the process of transcription and probably translation next time. But before that, there are more terms for you to remember to understand. Yeah. First of all, if you take all of your genetic material in a cell, that's called the genome. The genome. Okay? All your cells have the same set of DNA, right? I told you. Okay? So that's your genome. A gene, as I told you earlier today, is a section on a chromosome that contains the instruction to make a protein. So on a chromosome, you would have multiple genes. Okay, gene 1 is going to make protein 1, gene 2 is going to make protein 2, so on and so forth. These proteins, of course, have bodily functions, but they will also contribute to various features that you have. Okay, I keep on saying, like blood type, eye color, all these things are encoded within the gene, right? So the genome is like the entire textbook, okay, the whole textbook. Whereas chromosome would be like a chapter in the textbook. Okay, so we have 46 chromosomes, right? So it's like we have 46 chapters in this textbook. Within each chapter, like chapter 4, we will have chapter 4.1, 4.2, 4.3, right? And each subsection is going to teach you a particular concept. So those 4.1, 4.2, it's like a gene. You understand? Okay, so, chapter 22.5, that would be like chromosome 22, gene number 5. And finally, where you find a gene on the chromosome, it's called its locus. Locus would be like the page number. Okay, chromosome is really long, right? A bunch of ACTGs. Where does the gene begin and where does it end? Right? The exact location, that's called the locus of the gene. Okay, the location of the gene. Okay, genome, chromosome, gene, and locus. Any questions? So the chromosome 1 from mom and the chromosome 1 from dad, these two chromosomes are same length, same size, and they have the same kind of genetic information. Not exactly the same genetic information, but the same kind of genetic information. Okay? So we call them homologous chromosome. Homologous chromosome. So let me go back to our picture here, the one that we drew earlier today. Okay. And I'm just going to add to it for you. These two are homologous chromosomes. These two are homologous chromosomes. Okay. After you copy them, just take a look here. Again, these are sister chromatids, right? They are not homologous. They are identical. Okay. It's only homologous between the paternal and the maternal copy. Okay. Are these homologous? Yes or no? They are not, right? They are only homologous if they are the same chromosome number. Okay? If they are the same length and same size. 1 and 1 are homologous. 2 and 2 are homologous. So on and so forth, right? For females, XX are homologous. For male, X and Y are not because they're different. It's only homologous if they're the same. In chemistry, you learn about homogeneous mixture, right? Homogeneous means same, right? One phase. So that's what it means. It's the same. Same size, same length, same type of genetic information. Okay, so what do I mean by same type of genetic information? Well, over here, you might have, a gene for A, capital A. And over here, it might be the same gene. Right? If both of your parents have brown eyes, then you would inherit the brown eye gene, right? From both of them. This would be like, brown eye, brown eye. On another location, you might inherit like curly hair from one parent and then straight hair from another parent. I'm going to use a capital C here. So it's the same type of genetic information, hair type, but it's different version of that information, right? Depending on who your parents are. Right? Like obviously, even though your parents might be same ethnicity, they're not going to have all the same features, right? Okay. So like, do one more example. Like over here, one person might have dimple, and then the other will not have dimple. Dimples, those things that you have when you smile. That's actually genetic. Okay. So same order of genetic information, but the version of it might be different. They don't have to be different, but they might be different. So that's another word for you to learn. Different version of a gene is called allele. Allele. So like the brown eye, blue eye, that's different alleles. Okay. There could be many different alleles for a particular trait. Okay. Of course, it's not just brown eyes or blue eyes, right? There are like all kinds of eye color, right? Like green eyes, hazel eyes, whatever, right? So there will be multiple versions. Right? Are we together? Okay, it's this picture again where we talked about you started with DNA and then you make a copy of it in the form of RNA. That's called transcription. And then we talked about translation. But actually, there is a third step that I haven't been telling you about. Okay? After you do the transcription, it turns out the RNA that you created is not the final product. Okay? We have to change it a little bit, and that's called post-transcriptional modification. Okay, we're going to get into it. I just want to let you know that there is an extra step involved. All right. So why are we doing transcription? Why not just use the DNA and then make the protein directly? Well, I told you before, DNA is actually too big to leave the nucleus, right? It's not going to fit through the nuclear pores, so it's not going to be able to go out. And even if we are able to take the DNA out, it might not be the best idea because DNA can get damaged in the cytoplasm, okay? And so to prevent this problem, you make a copy of the gene that's needed in the form of mRNA. And more importantly, this adds a level of control. Right? Remember, all your cells have the same set of DNA, but not all your cells are going to need all the proteins that it encodes. Right? In the brain cells, maybe you only need, a group of 30 proteins out of the whole set. And then in the liver cell, maybe you only need 20 of them. Right? So which gene do you turn on and which gene do you turn off? You are able to control it by deciding what to transcribe and what not to transcribe. Okay. It's like a recipe book, right? You don't cook all the recipe in the entire book, even though you have it. You only cook the ones that you need or want, right? Yeah. So you only turn on the genes that you need. You should know by now, A pairs with T and G pairs with C, right? Yeah, I'd be a little bit disappointed if you don't know this by this point. The same rule applies when it comes to DNA to RNA pairing, right? Except in RNA, there are no thymine. Instead of T, we have the uracil, right? So A pairs with U, but G continues to pair with C. Okay, that's the only difference. Let's look at the process of transcription. Transcription takes place in the nucleus of a cell because that's where the DNA are. And the enzyme that does it is called RNA polymerase, which is what you would expect, because you're making RNA now instead of DNA. And there is no helicase or anything like that. The RNA polymerase is going to unzip the DNA. It's going to copy the DNA, and it will close the DNA. It does all that by itself. And in the end of the process, you create a pre-messenger RNA. Okay, the M stands for messenger. Now, this pre-messenger RNA is not ready for translation yet. Right? Like I told you, we have to process it before it can leave the nucleus. Right? It turns out there are some junk DNA in there that we need to remove, and it's not very stable, and so we need to take care of that as well. But first, let's take a closer look at the process of transcription. Right. You know what? Let's draw this out. So maybe it's going to make more sense to you. We have time. Okay, so you have your DNA here. And I'm just going to change it to a box so I don't have to keep on drawing the swigglies. But know that it is the squiggly, okay? And these are just a bunch of A's, C's, T's, and G's. Nothing more. So when you read a textbook, how do you know it's a new chapter? Like, I mean, it's all words, right? In it with some pictures, but how do you know it's a new chapter? Right? Yeah, like usually at the beginning of a chapter, there's like a title page, right? Almost, right? With some big pictures, and then it says um chapter 4 or something like that. So that's what happens. There's going to be a special sequence that marks the beginning of a gene, and that is called a promoter. Okay. AT-rich region So that's where the RNA polymerase is going to attach to. And that's where it's going to start the process of transcription. Okay, start copying here. That's basically what the promoter is telling it to do. And similarly, like how do you know you've reached the end of the chapter? Well, usually you're going to see the summary, you're going to see some practice question, right? Maybe a glossary and whatnot. Right, so here at the end of gene, we have a sequence that marks the end. It's called the terminator sequence. Now, I should make a side note here. It is AT rich because A and T, as you know, is weaker than C and G. So it makes it easier to unzip the DNA. Okay, so this whole thing, the promoter, the actual core of it, and the terminator, that is a gene. Okay, and then you might have some filler DNA before the next gene begins. Okay, basically, from one end to the chromosome to the other end, you're going to have a bunch of genes like that. Okay. All right, let's take a look at this next slide. The RNA polymerase unwinds the DNA strand and initiate messenger synthesis at the start point of the template strand. So basically, it binds to the promoter, and then it's going to start copying. Okay, that's what we just drew. All right, so that's initiation. Next step is elongation. Right. So as the RNA polymerase moves along the DNA, it will unzip the DNA ahead and rezip the DNA behind. Right? We're not trying to copy the entire DNA, so you need to close it up after you finish using it. Right? And this is where things get a little bit confusing. DNA have two strands, right? Okay. Only one of the two strands is going to contain the instruction to make the protein. Okay. The other half is only there for you to make a copy of. Okay. So this top strand here, we'll label it first, and then I will try to explain it. Oh, it's already labeled for you. This top strand here is the coding strand of the DNA. It contains the actual instruction for making the protein. This is the strand we try to copy. Okay, so the coding strand is the one we need. Remember what I told you before? I said if I asked you to copy ATTCG, you would just copy it as ATTCG, right? You would copy it directly, right? But in the body, whenever you try to make copies of nucleotides, the enzymes cannot copy directly. The enzyme will always make the complementary strand. So if it sees ATTCG, it will actually make like UAAGC. You understand? Enzymes always build the complementary strand. They cannot copy directly. And so if you want to make a copy of the coding strand, if you want to make this instruction, copy this instruction, you actually cannot use this to build the RNA. You will end up having the opposite of what you need. And that's what the template strand is for. Okay? The template strand is the strand that, I'll put it over here, is used for building the messenger RNA. So in this picture, you can see the coding strand and the template strands are complementary to each other. ATCA, and then you have TAGO. Actually, they probably made a mistake here with the, okay, never mind that. Okay, but they are complementary. But then when you build it, right, you're gonna build it off the template strand, so you end up having the same thing as the coding. Okay? I'll give you a better example when we come in the slides coming up. Okay? But that's the key thing to know. You cannot build the RNA using the coding strand because you will end up having opposite of what you need. The idea is similar to like minting a coin. Okay. If you want to make a dime in this case, actually this is that. No, this is a nickel. If you want to make a nickel, the mold that you use has to be a mirror image of what you want it to come out as, right? Right. See, this is backwards of the actual coin, right? You can see that. But once you add the metal to it and then you cast the coin, it's going to come out looking the way that it's supposed to be. You with me on that? Right? So that's what it is. If we want to build a coding strand, we cannot use the coding strand to build it because it's going to come out opposite. Right? So you're going to use the template strand so that when it comes out, it will be looking like what it's supposed to. Right? So it's kind of like that. So that process is going to continue until you reach the terminator sequence. Okay, and then you release the premature mRNA, and then the process of transcription is done. Okay, so what I want to do before we go is just 3 examples, and then we're gonna talk more about this on Wednesday. So these are the kind of stuff you are expected to do on the quiz. Right? Given the template strand, find the coding strand and the mRNA. Right? So this is template DNA. So what would be the coding DNA? If it's A on the template, what would be it? What would it be on the coding strand? T. Right. And then, yeah, it's just the complementary building pairing rule, right? Okay, so please fill in the rest. Okay, don't copy me, just do it yourself and then check it. What word am I going to use to describe the relationship? What's the relationship between template and coding strand? One word. Yep, complementary. Complementary means it follows base pairing rule, right? So now we're gonna do transcription. We're gonna make the RNA. So the RNA polymerase is gonna come, gonna open up a bubble, and then it's gonna build off the template strand. Okay? Watch. When the RNA polymerase comes, it sees A, what nucleotide is it gonna put for the RNA? U, that's it. Okay? Normally A pairs with T, but we are building RNA, so we're gonna have a U. Okay, so RNA polymerase is gonna move along. When it sees another A, it's gonna do U. What about C? What is it gonna build when it sees a C? Yeah. Continue. Sees a T is gonna put a A, right? And then so on and so forth, right? What's the relationship between the template DNA and messenger RNA? What word am I gonna use? Same word, right? Complementary. Because it also follows the base pairing rule, right? With one key exception, we don't have T's, so we're gonna use U, right? And I guess the important thing is this. In the end, if you look at the mRNA strand, it is actually identical to the coding strand, isn't it? Right? Remember, that's the one I want to make a copy of. Okay? It's identical with the exception that T becomes U. You understand that? Because there's no thymine in RNA. We are using uracil instead. So these two things are identical. Identical, but T becomes U. Okay? Essentially, it contains the same information. Please try example 2. Example 2 is this one. Now, this question does not tell you to create a coding, right? So you can actually jump directly to the messenger RNA. Okay. So the relationship between the template and the messenger RNA, what's the relationship? Complementary. Okay, so you just use the base pairing rule. A pairs with U, and then T pairs with A, and then the rest is easy. There will be a 5-mark question on this stuff on the quiz, okay? So make sure you know how to do this. Any questions? How about example 3? Create an mRNA from the given coding strand. Okay, this is coding. You have to make the go, right? Okay. Coding. Coding. If I give you the coding strand on the test, you should be very happy because it's very easy to change into mRNA. Remember, the mRNA you're trying to make is identical to coding, right? So you just copy everything with one exception. You have to change the T's to U's, right? So you see something like this, you just do it like that. You just copy it. Just copy it. Oops, sorry. Sometimes copying can be difficult. Okay. You just change all the T's to U's. Okay. Identical. Okay. I want you to understand it, of course, but if you can't understand it, then you can use the cheat sheet. You cannot bring it to the test, obviously, but you can use the cheat sheet while do your homework, okay? So this is coding. Coding is A, C, T, G, for example. Then what, because there are only 4 letters, right? Okay. What's the template, please? T, yeah. Right. What's the mRNA? I hear half the class say A, half the class said U. What is it?
You.
mRNA is same as coding, guys. The coding is the instruction, right? Okay, so it's gonna be A, right? But when you build it, when the RNA build it, it cannot build it from the coding because of the way it works, right? They don't know how to copy word for word. Every time you tell the enzyme to copy, it will only give you the complementary strand. Right? That's why you have to copy from the template. When it copies from the template, it switches it, right? So T is going to become A, which is good because it's the same as the coding, right? And then C, G is going to become C, right? So on and so forth. Understand? No? What's the question? How come is U there? Because this is coding, right? So coding and mRNA are the same. Coding and mRNA are the same. A is A, C is C, G is G, but T is going to become U because on RNA, there are no thymine, right? So that's the only difference. Right? So complementary. Right? Complementary. Identical. Okay. We have another row next time. All right, and then we have 4 more letters. So keep this somewhere safe. We're gonna add to it. Okay. So basically, this is a summary. And then you can go. Coding strand and mRNA are identical, except uracil replace all the thymine. Template strand and mRNA are complementary. And then you have the base pairing. Do not miss Wednesday's class. Right? Wednesday's class is very, very important. Right? If you don't know how to do the stuff that I teach you on Wednesday, you will not do well on the quiz. All right, that's it. Thank you, guys. Have a good one.